All right, chapter 22 continues our talk in biochemistry, and in particular, we're going to talk about um, nucleotides and nucleosides. Um, so, what a nucleic acid is, how they define it, is an unbranched polymer composed of repeating monomers called a nucleotide. All right, so there's a difference between a nucleoside and a nucleotide, which we'll talk about in the slides. There are two types of nucleic acids. One is our DNA and the other is RNA. DNA, or deoxyribonucleic acid, stores the genetic information of an organism and transmits that information from one generation to another, all right, which we kind of know. Okay, the RNA translates the code from the DNA into a protein. All right, so our DNA has the information, the RNA will take that information, and then it translates it into the actual proteins that our body uses. All right, so here's an example of a nucleotide, and this is a DNA molecule. The reason I know it's a DNA is that this carbon right here does not have an OH group. All right, and the D stands for deoxy. So DNA has one less OH group on that spot right there, where RNA does have the OH group there. All right, so basically it contains a sugar. In this case, our sugar is ribose. Okay, remember from the carbohydrate chapter, there's lots of different sugars. We basically talked mostly about fructose and glucose, but there's also a sugar called ribose, which this sugar is. That's why it's ribose nucleic acid or deoxyribose nucleic acid. A nitrogen containing base, there are several different ones. There are actually five. However, four of them are used in each of like the DNA and the RNA, and there's only one of them that is used in RNA that's not used in DNA. And there's one in DNA that's not used in RNA. And then finally, a phosphate group. Okay, if you have a phosphate group, so there's always three parts to it. A phosphate group, which they kind of sometimes show with a P in a circle, all right, and then you have your sugar, all right, and then you have that attached onto a base. Okay, um, the difference between a nucleotide and a nucleoside is a nucleotide includes all three of these: a phosphate, the sugar, and the base. A nucleoside is just the sugar and the base. So if you ever see a sugar, which I think there's a slide later on where they talk about that, and just the base, that's called a nucleoside. If you have the phosphate, it's a nucleotide. How I remembered it is in the word phosphate, there's a T, so it is nucleotide. That was kind of how I remembered it. All right, just some information about DNA and RNA. Most people usually know this. So DNA molecule contains several million nucleotides, while DNA molecules have only a few thousand. So our DNA is much, much, much larger than our RNA. DNA is contained in the chromosomes of the nucleus. Each chromosome have a different type of DNA. We have 26 chromosomes, 23 pairs, half from mother, the other half from a father. And a gene is the portion of the DNA molecule responsible for the synthesis of a single protein. All right. So in RNA, the monosaccharide is an aldopentose. It's D-ribose. All right, so here's the ribose sugar. In DNA, it's also a ribose. However, notice the OH is not there, hence the name deoxy. Now the bases. There are technically five types of our bases. DNA uses four and RNA use four of them. Okay. DNA uses, so these are the two, all depending on if you're talking RNA or DNA. The U we use in RNA, or we don't. Well, I guess we do technically. And the T is DNA. Okay, but all of us, but all depending on if it's RNA or DNA, we all use C, or DNA and RNA both use C, and they both use A and G. All right. This slide is just showing you that the C, the U, and the T use a pyrimidine as a parent, and purine is used for the A and the G, for adenine and guanine. All right, and in cytosine, uracil, and thiamine, they use a pyrimidine. Okay, so here they show in DNA it uses AGC and T, RNA uses AGC and U. So it's the thiamine and the uracil that are the difference between RNA and DNA. Okay, a nucleoside is formed by joining the anomeric carbon of the monosaccharide to the base. Okay, so let me see, they show a picture here. Yeah, they show a picture. Let me talk about this first. So remember, a nucleoside does not have the phosphate. The name a nucleoside derived from the pyrimidine base using a suffix idine. Okay, to name a nucleoside derived from a purine, you name it as osine. Okay, so for deoxyribonucleosides, add a prefix deoxy. All right, so they'll kind of show a name in here. Actually, let me lower this so you can see it. Okay, so remember the anomeric carbon. The anomeric carbon was the carbon that was the hemiacetal. Okay, you remember that? The hemiacetal? Hemiacetal. Okay, it's this carbon right here. All right, so what was special about that carbon? What makes something a hemiacetal? It's a carbon that's attached to two oxygens. All right, a carbon that's attached to two oxygens is what we call the anomeric carbon, okay, on a sugar. And that's a hemiacetal because one of them has a hydrogen on the oxygen and the other one has an R group or a carbon group. So it's called a hemiacetal. That's where the base always attaches to. All right, pretty much you form water. You see how this is going to form water. All right, so that goes away as water, and then the nitrogen itself bonds onto the anomeric carbon. So here's the molecule right here. Okay, so that is pretty much how that's formed, and then eventually a phosphate will come over and attach onto our nucleoside to form a nucleotide. All right, so yeah, okay. Hold on one second. Okay. I guess that here now they're just showing a picture of a deoxyribose with adenine to form deoxyadenosine. All right, deoxy. So this is what they were talking about the names, you know, earlier on the previous slides. All right, so here they're just showing an adenine hook up onto the anomeric carbon. All right, so nucleotides are formed by adding the phosphate group to the 5-OH. All right, when they name things, this carbon they call the 5 or the 5 prime, and then this carbon right here they usually call is the 3 prime because it's 5, 4, 3. All right, 2, 1. Our 1 is where our base is. Okay, so cytidine 5 monophosphate would be the name of this because that's because we're naming the base, plus at the 5th position we have a phosphate. So they do have abbreviations which make things a lot easier. Okay, so cytidine for the C, monophosphate. You guys have heard of ATP, right? Or ADP. We're going to actually talk about what those are. They are a type of a nucleotide. Okay, so actually here we go. Here is AMP, all right, or DAMP for deoxyadenosine 5 monophosphate. All right, so again, it's deoxyadenosine, all right, because it's a deoxyribose. We're using adenine, all right, but remember you name the name of it osine if you're using a purine, all right. If you're using a pyrimidine, it's idine. So this is a purine molecule. And that's one phosphate. All right. If you put 2 phosphates, it's ADP, adenosine 5 diphosphate. We usually don't say the number because the phosphates always attach onto that position. All right. And then lastly, we have ATP, adenosine 5 triphosphate, or ATP. All right. just to kind of give you some naming in there. All right, section 22.2, nucleic acids. Now let's start getting into the um, uh, uh, like the polymers of the nucleotide. So a nucleotide is a single unit. When you join multiple nucleotides altogether, you now have a nucleic acid, which is a polymer of nucleotides, and they join by the phosphate grouping from the 3 to the 5 position. So if you notice, here we had one of our nucleotides, and then we joined another one, and notice it joined at the OH group. So this is DNA. Okay. Notice the 3OH, the oxygen on the 3, joined up with the phosphate that's attached onto the 5. So it's always a 3-5 linkage. All right. So this phosphate now will join up with another nucleotide right here onto the 3 carbon, and so forth and so on. All right, and they call it a phosphodiester because you have a phosphate with a carbonyl with 2 oxygens attached to it, so it's a phosphate ester. All right, a polynucleotide contains a backbone consisting of alternating sugars and phosphate groups. The identity and order of the bases all distinguish one polynucleotide from another, the primary structure. And the polynucleotide has one free phosphate group at the 5 and one free OH at the 3. All right, and in DNA, the sequence of the bases carries the genetic information for the organism. Okay, so at the bottom of your chain, you have a free OH. That's what they're saying at the bottom of the chain. At the top of the chain, you always have the phosphate, the 5-phosphate. Okay, so let's see, the phosphodiester linkage joins the 3 carbon 1 nucleotide to the 5 of the other nucleotide. So that's what we just talked about. All right, this is kind of a neat little picture. I think I like how they write the word rather than the base, otherwise it gets very cluttered. And this is kind of a shorthand, like I was showing you a shorthand. They kind of show a phosphate, they kind of show the sugar in there, and then they show either the CATG. Obviously, if we had RNA, we would have OHs all at these positions, and instead of the T, we would have a U. Okay, so this polynucleotide would be called CATG, reading from the 5 end to the 3 end. Always read from the top end, or I'm sorry, the phosphate, the free phosphate end to the OH end. All right, some history about our DNA is actually a double helix. It's not a single strand, and it was proposed in 1953 by Watson and Crick. DNA consists of 2 polynucleotide strands that are wound up by right-handed double helix. The 2 strands run in opposite directions. So one runs from 5 end to the 3 end, and the other from the 3 to the 5. The sugar phosphate groups lie on the outside of the helix, and the bases lie on the inside. The bases always line up, so the pyrimidine derivative can hydrogen bond to a purine derivative on the other strand. That's how these are held all together. So I've got one strand. I'm going to try to make this as simple as possible. And then I have another strand, and you have hydrogen bonding that kind of hooks this all together. That's what holds it together. That's why hydrogen bonding is so incredibly important. I always tell my class, I don't remember if I've told this class, but if we didn't have hydrogen bonding, we'd just be a puddle of slime on the ground. We are just, everything is composed of hydrogen bonding. Thus, there are complementary base pairs that always hydrogen bond together. We'll talk about that. The G and the C always hydrogen bond, and the A and the T always hydrogen bond, or the A and the U if we're talking about RNA. Okay, so here's the slide. I just gave you a heads up. Adenine pairs up with thymine with 2 hydrogen bonds to form an AT base pair, and cysteine pairs up with guanine using 3 hydrogen bonds to form a CG pair. Okay, again, that's in DNA. Do they show RNA? But RNA instead of T would be U. Okay, so and also remember it's always opposite. It's 5 to 3 to 3 to 5. All right. All right, so here's just an example of a double helix, and they're kind of showing you how G and C are bonded to each other, and A and T are bonded to each other. And this is what's happening. You have these hydrogen bonds um, with our groups. The hydrogen bonds fit perfectly with the groups we have. All right, kind of interesting how that happened. All right, so the GC pair has 3 hydrogen bonds, and the AT pair only has 2 hydrogen bonds. All right. The information stored in DNA is used to direct the synthesis of protein. So there's what's called a codon. I think it's every 3 base pairs is for 1 amino acid. So like a sequence will tell you your body, okay, this sequence says we need this amino acid. Then the next 3 says we need this amino acid, and then it hooks it all together. It talks about it later on. Replication is the process by which DNA makes a copy of itself within a cell. Transcription is the ordered synthesis of RNA from DNA. The genetic information stored in DNA is passed onto the RNA. And then translation is the synthesis of the proteins from the RNA. The genetic information determined to determine the specific amino acid sequence of proteins. All right, so the DNA makes the RNA then goes ahead and makes the proteins. So here's kind of the order of it. You have our DNA, our parent DNA, and when it replicates itself, it makes a daughter DNA, all right, which then it becomes the parent DNA eventually. But the parent DNA, you do transcription to make RNA, and then you translate the RNA to finally make a protein. All right, big, huge process. All right, so we'll stop it right there and start back with the next video.